Radhe Radhe everyone, welcome to my YouTube channel Biology with Tricks. So sorry guys, I haven't uploaded any video in past few days because I am little bit busy. But now onwards I'll be able or I'll be provide you some important topic notes with some lectures related to CSIR NET. Before any further delay, today I'm gonna talk about apoptosis and C.Elegans, right? It's a most important topic and you guys will get 2 to 3 questions from this topic only, and this question will be of part C. And you all guys know who are preparing for the CSIR NET exam, part C each question carries a 4 marks, right? Means if you got a 2 question from here, you can get a 8 marks, right? And this topic is a short topic and I'll give you a precise data and it will be beneficial for you guys, right? Okay, we'll start with the apoptosis in C.Elegans. Before starting with the genes, I'll just want to give you a basic information. C.Elegan is basically a hermaphrodite. Hermaphrodite means it has a both sex organs, female sex organs and male sex organs, right? And its nature is depend upon also temperature. At 16 degrees it can live at 16 degrees it can live about 2 to 3 days, and at 20 degrees it can live about 3 to 4 days, and 25 degrees it can live about 4 to 5 days. This is about the basic information of the C.Elegan. Now we'll starting with the apoptosis in C.Elegan. There is a presence of some genes, basically CD3, CD4, CD9 and EGL1. Basically CSIR NET not directly ask a question from here. They will ask the homology between these genes and the genes in a human which are playing a role in apoptosis, right? Human BCL2 is a analog of or homology of the CD9, right? It is a similar, homology means similar, it is performing a similar function like a CD9, right? We need to focus on this diagram. This is showing the which genes are correlated with each other. CD9 is correlated with BCL2 and vice versa. CD4 is performing a function like APAF1 in humans, and CD3 is equal to caspase 9, right? And this diagram was also showing that CD9 is inhibiting the CD4 function and it will not going to apoptotic or cell death, right? It means if CD9 is inactivated, then it will leads to the apoptosis of a cell. If it is activated, then it will lead to the inactivation of CD4 and there is a no cell death. We can conclude it from here that analogous genes CD4 APAF1 is a protease activating factor which will lead to the breakdown of the CD3, resulting into the activation of the CD3 caspase 9, which will initiate the cell death, right? In case of the C.Elegan, there is a presence of EGL1 which is inhibiting a CD9, right? If CD9 is inactivated, it will which is inhibiting the CD4, this will result into the activation of CD4. Now this CD4 get activated and result into the proteolytic breakdown of the CD3, resulting into the activation of the caspase 9, which will result into the apoptosis. Similar case of humans, BIM BH3 inhibiting the BCL2, which result into the activation of back and backs. This result into the release of the cytochrome C, right? These cytochrome C bind to the APAF1, which is equivalent to the CD4 in C.Elegans, and this APAF1 activates the caspase 9 and resulting to the activation of caspase 3 and proteolytic activity of the cell, right? This is about the basic information. CSIR basically asks a question that which gene in C.Elegan is analogous to which gene in humans, right? Like CD9 is equivalent to the BCL2, CD4 is equivalent to the APAF1 and CD3 is equivalent to the caspase 9. This is about the basic information. And there are two important statements that I want to tell you that basically CD9 gain of function and loss of function in CD4, this will result into having a 947 non-gonadal cells, while CD8 is a multi-spanning plasma membrane protein that basically require for externalization of the phosphatidylserine. This is we also seen in case of apoptosis in mammals that caspases require a phosphatidylserine outside a membrane or externally. In case of C.Elegan it is also similar, but it is showing about the CD8 function. This is about basics, that these two important statements must asked in CSIR NET exam, because you will get a statement type question and you have to choose a one correct option, right? And these two options may be one of them, and I am pretty sure you'll get a 2 to 3 question from this topic. And the people who are preparing for CSIR NET exam must know that in part C questions carry 4 marks, and if you got a question from here about 2 to 3, you will be able to secure a 8 to 12 marks. Okay, so don't be too lenient, please read out this topic. I'm just giving you the precise version of this topic, the most important points from where the question has been asked and will be asked in July 2025 and December 2025 also. Okay, after that I am just giving that inhibitors of apoptosis. There are some inhibitors of apoptosis. We call it as a IAPs. They are a class of protein that known as the regulators of the caspases and immune signaling, right? They are also known as the BIRC, baculovirus inhibitors, or domain containing proteins characterized by the presence of baculovirus IAP repeats. Okay, they are zinc ion coordinates. They are highly conserved from virus to mammal, and metacaspase are the homologue of caspase, result to programmed cell death in yeast and plants. Basically, in yeast and plants, there are the metacaspases which are equivalent to the caspases, result into the apoptosis of the cell, right? Basically, there are 8 types of the IAPs, right? BIRC1, BIRC2, BIRC3, similar to 4, 6, 7, 8. BIRC1, there is CSI net must ask a question which BIRC is equivalent to what function. BIRC1 is performing function in a neuronal, BIRC2 in cellular level function, BIRC3 also in a cellular level, and BIRC4 X-linked. While the 5 and 6 not yet been so much discussed, but BIRC7 in melanoma, which is a cancer, and BIRC8 in IAP like proteins, right? These they function as a blocking of caspase 3. They function by blocking caspase 3, 7 and 9, right? There are some anti-IAPs in Drosophila which is resulting into the inactivation of the IAPs, right? These are genes rapper, hit and grain. These genes activated cell death by inhibiting the anti-apoptotic activity of Drosophila, while gain of function of mutation in the Drosophila, which is also known as DIAP1, strongly suppress the rapper, hidden grain, so no apoptosis. As I said that these are the genes which are resulting into the apoptosis. If the Drosophila mutation in DIAP is, there is a no apoptosis. Then these are the two important points, right? That mutation in CD9, which is inhibiting the CD4. If the mutation occurs, CD4 always get active, and this will result into the death of the cell. If there is a gain of function of CD9, it result into the inhibiting of CD4 and the cell do not die, they'll survive. This is about the importance of the apoptosis in a C.Elegan, and I hope you guys will like it.